So I hope the idea how the signal is transduced, the light is transduced, is fairly clear. Okay, now sort of what happens during the, so you know how you walk into the darkness and you can't see anything first, and then your eyes adapt. Or opposite, you walk from the dark room into the light, and for a minute you feel like you blinded. So we're going to chat about molecular mechanisms behind it. Okay? Now, what happens when you are stepping out in the bright light? So first, since it's a bright light, there's a strong stimulation of the opsin, of the photopigments, both cones and rods. Does that make sense? What about rods? Are they sensitive? They're extremely sensitive. They're extremely sensitive to the light. So for them, remember we talked about receptors, the sensory perception, and we mentioned one of the conditions for the sensory perception is that stimulus is in the sensory range, perception range. For rods, the bright light is way out of range. It's way beyond their capacity to absorb light. Okay? So what happens? They are bleached out immediately. All rhodopsins dissociate. Okay, so opsin component and retinal component dissociate. Think about this, there's light all the time, so they are always dissociated. Does that make sense? So opsin component goes into the inner segment, it translocates. There is no opsin in the outer segment, in those discs where photoreception occurs, so there's no photoreception from rods. Does that make sense? All rods are bleached out. Okay? Then cones adapt to the brightness, which increases the sensitivity of retina, and you can see in the light. Does that make sense? Now you going back from the bright light to the dark. Immediately after you enter the dark room, two things are going on. Thing number one, your cones do not have enough sensitivity. Does that make sense? Do you understand what I'm saying? Cones do not have enough sensitivity, so they stop signaling. This time, the stimulus is out of their perception range. Okay? The only difference is that now the stimulus is too low for the perception range. And rods are not functional yet. Remember we mentioned that rods are bleached. All the opsin is in the inner segment. They are dissociated with retinal. So it's a situation like cones just, throw the towel and say, I'm out. And rods are not even ready for the perception. That's why you are completely blinded when you step into the dark room. You cannot see a squat. Okay? Eventually. Since now it is dark, rhodopsin will start to accumulate. Does that make sense? This process will happen. All trans opsin will be converted into the 11-cis. Sorry, all trans retinal will be converted into the 11-cis retinal. 11-cis retinal will bind to the opsin component, and you're going to have your photopigment. Okay? As photopigment accumulate, transducin, the G protein, can go back to the segment. Oh, that's some wild stuff happens here. Okay, so you have functional photopigment accumulating in the outer segment, and again you start seeing things. Does that make sense? We good? Is that understood? Now, what can affect your night vision? Your night vision is dependent on rods, right? Cones have no role in it. Does that make sense? And some people, especially people with the deficit of vitamin A in the diet, rods cannot perceive the light. Is that clear? Because lack of sufficient amounts of vitamin A leads to the decreased amount of 11-cis retinal. Here's the thing you have to appreciate. Remember how we talked about the recycling? Pigment cells can recycle fragments of the photoreceptor cells. Let's be honest, in any process of recycling, it's not 100% efficient. So the damage that is done to photoreceptor cells, some of that retinal will be lost, chemically destroyed. Okay, so you need a source to replenish your retinal supply. Does that make sense? That's what vitamin A is for. If people do not get enough vitamin A, the amount of retinal decreases, the amount of functional rhodopsin decreases, and your perception of the light in the dark room, your night vision will decrease. That picture in the left bottom corner, I know it's not very precise, but you can sort of appreciate on the left. That's what the person with normal vision sees in the night. And the person with the night blindness will see this. Still be something, but not as sharp, not as detailed as it would be in a person with normal vision. Does that make sense? So in developing countries, the condition, the night blindness, is often associated with a deficit of vitamin A, which can be found in like carrots and other stuff. It's fat-soluble vitamin, so if you eat your carrots, eat them with like sour cream or any other oily substance to increase the absorption of that fat-soluble vitamin. But generally, if you would look at the label on any product that you buy in the United States, practically any, almost everything is enriched in two vitamins, vitamin A and vitamin C. Okay? So you get enough vitamin A with your diet. In developed countries, the problem lies in a different problems different. People with night blindness in developed countries most often have a condition called Retinitis Pigmentosa, which is the fancy name for dysfunctional pigment cells. When pigment cells cannot properly recycle photoreceptors, the amount of retinal that is returned into the photoreceptor cells is practically zero. So loss of retinal is enormous. A supply of vitamin A cannot possibly substitute for this loss, and people slowly lose the night vision. Make sense? Small factoid, that same disease in Russian is called not night blindness, but chicken blindness. No idea. Chicken blindness. Okay. So hopefully we figured out how the light is transduced into the nerve signal. Now what happens then to those nerve signals. They go through the optic nerves, which consist of several, bunches of fibers, consist of all those fascicles, and those bunches, they separate at the optic chiasma. In this picture, you can see that one bunch goes into the opposite hemisphere, contralateral hemisphere, and another bunch goes into the ipsilateral hemisphere. Does that make sense so far? Optic nerves here no idea what that is. Optic nerves, they go from the eye to the optic chiasma. After optic chiasma, the structures that reach brain called optic tracts. Okay, so that's optic tract. Does it make sense? This is really a fundamental thing, fundamental concept. The fibers from each eye go to both hemispheres. Conversely, you can see that each hemisphere receives the signals from each eye. Does it make sense? Your left hemisphere receives signals from both left and right eye. Same goes for the right hemisphere. Why it is so important? This is retina. That's the perception area of the retina. So it means that this is your visual field. And a huge part of that visual field will be seen with both eyes. Some peripheral areas will be seen with right or left eye only. And you can see that if you close one eye, you will see how the part of the periphery just disappears. You close left eye, and the part of the periphery disappears. You close right eye, and the part of the periphery disappears. And it's absolutely logical, seriously, okay? Because something that can be seen with only right eye or left eye or whatever. Does that make sense? So, what I wanted to say. That overlap is about depth perception, and we'll talk about it in a second. Now, in terms of the pathways, these optic tracts, they first reach the thalamus. And then from the thalamus, output goes into the visual cortex. Well, output, input, whatever. Sensory signals are related to the visual cortex. Does that make sense? So that part, those fibers that go to the visual cortex in the occipital lobe, will be responsible for your conscious perception of whatever you see. It's going to be primary and association cortices that will tell, who you are seeing. Okay, what you are seeing. Is that red, blue, or green? You consciously perceive. Do you recognize it? Okay, all that kind of stuff. Some fibers will reach to the midbrain. Okay, right here in the superior colliculi. This part of the midbrain controls the muscles of the eye. So remember we talked about following object, the visual reflex. That's the visual reflex. Does that make sense? So for instance, if I will start, playing ping pong with someone, you will all be doing like this. That's the visual reflex that's controlled by midbrain. Because you see moving object. Right? And another important input, hypothalamus. Pupillary reflexes, constriction and dilation of the pupil in response to the light, and circadian rhythms. I don't remember if we talked about circadian rhythms. Huh? Yeah, well, here's the point. light is an important regulator of your sleep-wake cycle. Okay, period. The mechanism is elegant, though. It signals to the pineal gland, which produces melatonin, and when there's a light, production of melatonin goes down. When there's dark, production of melatonin goes up. When melatonin is increased, it stimulates sleep. One of the precursors for melatonin is serotonin. So that is one of the few nutritional supplements that you can buy over the counter that actually works. Melatonin, you pop the pill and you go to sleep, and you sleep really well. It is especially useful in the conditions where your circadian rhythms and daylight cycle is changed, okay? Not synchronized when you fly like overseas. And it is useful if you work far beyond the polar circle, where the day and night cycles are completely screwed up. And maybe I mentioned that one of my friends spent 2 weeks in very up north, in the conditions that are called white nights. I mean, it's not even a night, it's pretty much a polar day. So there is no night whatsoever. There's a little bit of like evening, I would say, twilight, but that's about it. And she said that although she knew what time it is exactly, she couldn't go to sleep. It could be 11 p.m. It's like daylight out there. So your circadian rhythm is completely thrown off. And by the way, in animal colonies, in experimental institutions, there's a timer that is set to turn on and turn off the lights, because animals have circadian rhythms too. Does that make sense? So it's kind of interesting, because mice are generally nocturnal animals. When light is off, they run around, and then if you do something late at night, you walk in there 11 p.m. in the evening, and you turn on the light so you can work, and they just, hey, what's going on? Everybody's like, start, light is everywhere. So you kind of mess it up a little bit, okay? Go ahead. Completely backwards. It's a great question. Completely switched, I suppose, yes. It's going to be a pathology. Technically, the shifted circadian rhythms are considered to be a sleep pathology. There are people that are comfortably going to bed at 3, 4 a.m. in the morning, but then if, there's no call of duty, they sleep till 1, 2 p.m. in the afternoon. Technically, it's an anomaly. It's not normal circadian rhythms. But I would say that this is probably the least of the problems that you can think of. Just general, If the person doesn't have to wake up at 8 a.m. in the morning to go to work, then let the person sleep. So technically, I suppose there can be a situation when the person goes to sleep at 8 a.m. in the morning, wake up at 5, 6, 7 p.m. in the evening. And speaking of which, again, it's not, science works with large numbers. So one of my buddies when I was in Louisiana, he was in grad school. He's from Jordan. During the month of Ramadan, you cannot eat during the night. During the day, you cannot drink a lot of them don't drink during the day. But then, believe me, the night starts with festivities. It's really fun in the night. So he said that for him it's really hard because back home, he said, people just took like Ramadan off and they slept through the day. And then they were partying during the night, you know, and then they came home before the dawn and just slept through the entire day, and then after dusk they just go out and have fun. Okay. I worked night shifts, but I also had to be in college during the daytime, so pretty much I had like no sleep. It's hard. The severity depends. Some people don't drink water. Okay, but yeah, depends. Now, say again? Yeah, well, there's a lot of restrictions on that. So, central part of the visual field, okay, gets to the lateral part of the retina. It's kind of exemplified here. So you see the yellow part goes to the lateral. The blue part goes to the lateral. Okay? And medial part of the retina looks at the temporal region of the visual field. Now, fields overlap. That enables us the depth perception, to have depth perception. Does it make sense? Depth perception is because of two things. First, having two slightly shifted images. Have you ever watched 3D movie? Have you ever tried to watch 3D movie without 3D glasses? How does it look? Fuzzy. In 3D, I'm not extremely familiar with the technology, but essentially I have two pictures projected on the screen that are slightly shifted against each other. Wearing those glasses, since there are two pictures, it adds 3D component to the image on the screen. So, overlapping images that you see with both eyes make you aware of the depth. That's why if I throw the ball to Jacques, you know, I'm not going to hit Esther, right? Because hopefully. So I know how far should I throw. Does that make sense? If the person loses the eye due to the trauma or accident or something, eye loss completely and totally eliminates depth perception because there is no overlap. Do you see what I'm trying to say? Great question. Close your eye. 1. Do you still understand that I'm farther than James? Yes. Why? Because you know. Environmental cues. You know. Like, if I close one eye, I still know that Alexis is closer than the door. Just because I have so many environmental cues that I can consciously process. But if you would ever try to pitch a baseball, play, I don't know, tennis with one eye closed, you will soon notice your inferiority. Okay? Because that influences your movements. That's your cerebellum that refines your motor activities. You're playing sports. It won't have that cue. Does that make sense? Now, that's interesting. So, people who are one eye blind, they don't have depth perception. Okay? However, if there is a loss of vision behind the optic chiasma, not in the tract, sorry, not in the nerve, but in the tract, depth perception is not lost. Because there is, think about this, and I'm going to explain why. If there is a damage right here, are both eyes functionally good? Eyes, Eyes. Eyes themselves are completely functional. Does that make sense? They're good. They're fine. Does every impulse, every signal from eyes reach the brain? No. But there is an overlap. That essentially those yellow fibers from both eyes will reach the hemisphere. That makes sense? Which means there's going to be some overlap of the visual fields. Some, not complete, but some. And that some overlap will provide the degree of depth perception. What's going to be impaired in the tract damage? The visual area, right? So the width of the visual field will be decreased in the tract damage, but depth perception will remain. Does that make sense?
Yes. We were covering the strabismus.
Yes.
Not long ago, and is treatments for
Yep.
that. So, in a sense, is the patching doing damage as well as good?
It doesn't do permanent damage. It doesn't. Because what happens, that's a good question. So kids who wear those patches over their eye to correct for lazy eye, for strabismus, if it is caused by the weak eye muscles, essentially they become kind of sort of one eye blind, and they lose the depth perception, but they lose depth perception for the time of training. Does that make sense? So no, it's not permanent damage. Permanent damage is only when like eye is gone, totally gone. Does that make sense? And that may, you may see it. What can damage, what do you think, what can damage the optic tract? Mostly stroke. Yep. It's mostly stroke. So stroke may affect somebody's vision pretty severely. Optic nerve as well, especially if it's anterior cerebral artery. Anterior cerebral arteries, they somewhere in this ballpark, so that may affect somebody's vision. Does that make sense? Now, in terms of the picture processing, it goes through several steps. So in the retina, it's a simplification and condensing of information. Depends on, cones and roads, they're all going to do that. And they will allow you, think about this, color. What's responsible for color? Three different types of pigments, right? They spectral overlap, that's why we see color. Brightness. Change in brightness, what will it change? Why do you know that, like, not only this, why do you know that this is brighter than this? What happens at the molecular level in your bipolar ganglion cells? Well, in order to get transmitted, photoreceptor cell is hyperpolarized and bipolar is depolarized. But what happens as the result of bipolar cell depolarization? What does it release? The neurotransmitter. The brighter the light, the more neurotransmitter is released. Does that make sense? More neurotransmitter, higher frequency of action potentials. Does it make sense? And then, retina will be the first step to identify, the speed of movement, the direction of movement, the angle of movement, and all that kind of stuff. And then, thalamus is responsible for that visual fields overlapping and depth perception. Also increases the contrast and emphasizes the input of cones if there is any. And primary cortex, contrast in which way object is, upside down, downside up, you know. And then processes, so those impulses regarding the motion of the object or color of the object are to visual cortices, visual association cortices. In association cortices, that's where you identify objects, okay? That's where you relate objects to others in space. So essentially, going back to our idea about not completely losing depth perception when one eye is closed, you still know that this computer screen is in front of me, because you know if you can't see part of me, it must be in front. Does that make sense? So those cues come from all that experience that we had over time.
So this kind of seems to give an explanation as to why people are taught to shoot. Like some people are taught to shoot one eye closed, and then like, you know, as you get more comfortable, you're taught to shoot with both eyes, like for that depth perception. People with one eye shoot better on paper targets, like just kind of numbers wise than they do when you're actually in the field.
Probably so, because well, I don't know the statistics on the field. It would be hard to collect because you would need a lot of experimental subjects like humans that you will shoot, which would be hugely unethical. I'm just kidding. No, what I'm saying is it would be interesting to compare the performance of one-eyed shooters in terms of the, you know, standing target, doesn't matter if it's paper or not, versus moving targets, and moving not only laterally but also anterior and posterior movement. Because if they are unable to process that movement, or they are inferior in that processing, they wouldn't be so good in shooting something that goes like this direction. Does that make sense? I don't know about the data, but that would be interesting to learn. Okay. Any questions? What I want to do with you now, we're probably going to start here, but I wanted just to chat about something else. Light transduction seems pretty important and pretty hard. It's hardcore physiology. So how about you're going to guide me? I'm going to ask you questions, what happens, and you tell me. And if you can skip the PowerPoint and just do it like as the hive mind, collective brain, you know, that would be great. Okay? Try to participate more actively in our discussion. Does that make sense? So we're going to start with what absorbs light? Tell me. Only rods. What else absorbs light? Huh? No, they refract. They refract. What absorbs light? Which two types of photoreceptor cells? Rods and cones. Okay. Now, what in the rods and in the cones absorbs light? Molecules. See again? Okay, 11 says photopigment. And the component of photopigment that is changed in response to light is 11 cis retinal. So what is photopigment anyway? What does it consist of? Okay, opsin is what? Chemically? Protein, yeah. Okay, 11 cis, as Jessica says there. When there is light, what happens? When there is light. What happens to 11 cis retinal? It becomes what? All trans and separates from opsin. So that idea makes sense. So this process seems rather simple, right? Seriously, it's just simple. Okay. Awesome. Ooh, it's a field day. Now, let's do the transduction. Okay, so we're going to start with the membrane and that opsin thing, that protein. Is it peripheral or transmembrane? Transmembrane. And what is it bound to? Okay. So when light interacts with 11 cis retinal, 11 cis becomes all trans. There you go. So now we have, and it dissociates. Sorry, it dissociated the wrong way. Should dissociate in the cytoplasm. On the cytoplasmic side of the opsin, there's a protein which is named with one letter, G protein. When all transretinal dissociates from opsin, what happens to the G protein? It is activated, and we're going to use asterisk for activation. And as it is activated, what does it do next? It binds to the enzyme, and it's just whatever. And does what to the enzyme? Activates the enzyme. How do we call the enzyme? Phosphodiesterase. Phosphodiesterase, and it is activated now. What does phosphodiesterase do? Say it one more time. Okay. Breaks down cGMP, producing just GMP. Never told you what GMP is, actually. It's guanosine monophosphate. Have you heard about ATP? It's adenosine triphosphate. ADP, adenosine diphosphate, and AMP, adenosine monophosphate. It's exactly just same pattern. So GMP is guanosine monophosphate. What do you think C stands for? Cyclic. It has a cycle. When the cycle is broken, cyclic GMP becomes just GMP. Now, what's the function of cyclic GMP? And what flows through those channels? So when cGMP is destroyed, what happens to the channels? Any flow? When the flow is stopped, what happens to the membrane potential of the cell? Decreases, making the cell oh, wait, no. Now, all what we just discussed, where did it happen? In which cells? photoreceptor cells, which is connected to what cell? Which is connected to Does that make sense so far? When there is no light, which neurotransmitters are produced and where? Inhibitory neurotransmitters produced where? Photoreceptor cell produces inhibitory neurotransmitter. Somebody else. It's Alexa's solo performance. Somebody else. Come on. Bipolar cell. Does it produce anything? No. No, it doesn't. No light. Inhibitory neurotransmitter. What does it do to bipolar cell? In terms of polarization. Bipolar cell is hyperpolarized. No neurotransmitter, right? And there is light. What happens? Photoreceptor cell is... Photoreceptor cell is when there is light. It is hyperpolarized. Does it make sense? Light essentially leads to the destruction of cyclic GMP, closure of the channels, no positive ion influx into the cell, and hyperpolarization of the photoreceptor cell. Okay? If it's hyperpolarized, what happens to inhibitory neurotransmitter? It is not produced, right? What happens to the bipolar cell? And what does it start to churn out? Excitatory. Very good. Excitatory neurotransmitter. And you've got action potential. Does that make sense? Any questions? You can try my suggestion, my personal opinion, how to do like the studying for this. That very first graph that we did with like membrane, enzymes and everything, just try to draw it. And as I mentioned before, you probably will find that you're missing things in your very first try. And then you do it on several occasions, you're going to be just fine. And same here, Once you know that light hyperpolarizes photoreceptor cell, you can go from there. That thing that we played with, like, photoreceptor cell holding bipolar cell back, I consider it one of the most important concepts that we cover in the vision. And it's also, I wouldn't say the hardest, but arguably, you have to understand which neurotransmitter is produced where. Does that make sense? Okay, we're gonna start chatting about the ear, and then in about 10 minutes I will run downstairs, grab the quiz, and bring it back. Sounds good? Ear, 3 parts. Actually, let me pause it. Has 3 parts. External, and I didn't get one for myself. Never mind. Try to identify it yourself. External, which is pretty much, which I can show myself. That's external ear. Okay? Middle ear and the internal or inner ear. Outside, the external part is pretty boring. It's auricle, which is essentially an elastic cartilage covered by skin. The main function of the auricle is to, it is shaped a little bit like a spiral, so it channels the sound into the tube that is called external acoustic meatus. External acoustic meatus is essentially a hole in the temporal bone of the skull, and if you would look at Marvin, you will be able to see a little bit of external acoustic meatus in the temporal bone. I can't reach him. I can't reach anything here. God damn it! And I don't have a skull. Maybe I do. Thank you. So this is our friend Marvin. You can see external acoustic meatus right here. Okay, in the temporal bone. That's where the sound goes to. It's actually a physiologically important structure. Hair in the external acoustic meatus, they collect and trap dust and dirt and microbes actually. There's a bunch of sebaceous glands, of course, it's a skin, and ceruminous glands. So your ears, they don't sweat. They produce wax. Ceruminous glands produce wax. Wax plays another important, pretty much the same important function. It traps dust and dirt. Should you clear the ear? That's a hard question. Well, probably should wash them from time to time. But one of ear, nose, and throat doctors that I personally know, he has his favorite saying: nothing smaller than the elbow should go into your ear, which pretty much means keep your fingers away from it, because the tympanic membrane that acoustic meatus ends with is fairly easily damaged. Okay, so that tympanic membrane right here is the transparent membrane that forms essentially a boundary between the middle and outer ear. The membrane consists of the connective tissue, and its function is to convey the vibration, the air vibration, which is sound, that come through the external acoustic meatus to the structures of the middle ear. On the outside, tympanic membrane is covered by the skin, and on the inside by the mucosal layer. And when air, the vibrating air, starts to vibrate tympanic membrane, vibration of the tympanic membrane right here are transmitted to the bones of the middle ear called auditory ossicles. Okay? Inside of the middle ear there are two openings. One is called oval window, one is called round window. Oval window is immediately under one of auditory ossicles. Round window is by itself at the entrance of pharyngotympanic tube. These two windows provide a certain level of mechanical communication between the middle and inner ear. Let's recall the movement of sound of those mechanical vibrations that we just talked about. So sound first, sound first is channeled by, what was that? Gotta be kidding me. Sound first is channeled by the auricle into the external acoustic meatus. It reaches tympanic membrane. Vibrations go through the auditory ossicles and through the oval window directly in the inner ear. We didn't talk about inner ear yet. It's not time yet. Does that make sense? Structure and functions and all that. Yes. When you do this, that's tympanic membrane. Ugh, that pops. Okay. The middle ear and your pharynx, throat, are connected by the tube called pharyngotympanic or Eustachian tube. It goes directly into nasopharynx, the upper portion of the throat. And that's why those doctors called ear, nose, and throat, all those parts are anatomically connected. You probably noticed, if you start to laugh when you eat, the food from your throat can go into your nose. I saw with these own eyes a poor boy who had ear infection and got his ears washed, and it looked pretty horrible. It was, I'm not kidding, I saw it. I was like 9-year-old, I was scared shitless. Huge syringe without the needle, and they were just pushing the disinfection fluid through the ear, and it was coming out of the boy's nose. It's pretty bad. So they all connected, and that's important. Does that make sense? Go ahead. No, he had pierced tympanic membrane. That sometimes we'll talk about it in a second. The myringotomy, right here, in the upper right corner. This one. Does that make sense? So normally Eustachian tube is closed. Every time we yawn, every time we swallow, it opens and it equilibrates the pressure outside of us and inside of the middle ear. Now think for a second. This is essentially outside. Let me go back for a second. This is outside. Does it make sense? This is middle ear. If the pressure on the outside is lower, what's going to happen to the membrane? That's outside. This pressure is higher. It's going to do this, right? Does that make sense? If it's the opposite, it's going to kind of do a little bit more, into the ear, bulge into the ear. The difference in pressure will actually put severe restriction on the membrane vibration because it will be more tense under the pressure. Does that make sense? By opening the Eustachian tube, we equalize the pressure here and make membrane, normally loose. Does that make sense? So every time, as you mentioned, when you do this, that ear pop is because you force air into the ears. Okay? And technically, hypothetically, if you have pierced tympanic membrane and you do it really well underwater, there are going to be bubbles coming out of your ears. Okay? The fact that the throat, nose and throat, nasopharynx, is connected to the ear determines why people can get otitis media, the infection of the middle ear, especially kids. In children, this tube is more horizontal than in adults. So snot, if kids have like a throat infection or nasal infection, snot can reach middle ear, or it just can be flow of mucus from the nasopharynx, But if this mucus contains any pathogens, mostly bacteria, different types, Staph aureus, Streptococcus, strep throat, strep pyogenes, Hib, Haemophilus influenza, it reaches the middle ear and causes inflammation. Inflammation leads to the accumulation of fluid. That leads to the bulging of the eardrum. And if bulging is severe enough, eardrum may rupture. Otitis media is the leading cause of hearing loss in children, not in newborns. That's different. In newborns, it's different. Okay? But in kids, it's a leading cause of hearing loss. Do you see the mechanism? Now, what is the treatment? If you have middle ear infection, that shows you, the difference. That's normal eardrum. You see normal color. That's the inflamed one. And you can kind of see the pus that accumulates behind the eardrum in the middle ear. Yes? And they usually don't swim in sterile waters, so they contain a lot of microorganisms which can cause infection, yes. Now, if anyone had otitis media, I suppose yes, you don't have to answer. What do you get when you go to the doctor? What do they prescribe? Antibiotics, right? Here's the interesting thing. The necessity of antibiotics is questionable because 81% cases, in some studies up to 81% cases of otitis media recover by themselves without antibiotics. I read a study in which doctors show, it's all studies, you have to understand, it's like maybe hundreds or thousands of kids, but they demonstrated that antibiotics, they do improve clinical outcomes. They had certain criteria. 14% better. If you want to translate it to something, more reasonable, one study showed that use of antibiotics in case of middle ear infection shortens the time the symptoms persist by 12 hours. Instead of 6 days, it's 5.5, for instance. Which is terrible. And now with more and more antibiotic-resistant bacteria, many physicians question, do we really need to give antibiotics to a child? Or we can observe, and if it becomes worse, then we do the intervention. Does that make sense? So there is no, agreement. Some kids get middle ear infection very frequently. Very frequently. In this case, myringotomy is warranted. Myringotomy is the piercing of the tympanic membrane and introduction of the tubes into the tympanic membrane. It's a relatively quick procedure. It takes about 2 hours, including anesthesia. Okay? And these tubes allow the unrestricted flow of pus out of the middle ear, if there is any. Okay? And then as the child grows, two things happen. First, pharyngotympanic tube becomes less and less horizontal and more vertical. And two, tympanic membrane grows as well, and those tubes fall out by themselves. Oh, I know a few people who had that. That's pretty common. And that's a great treatment because that sort of eliminates the necessity in, giving kid antibiotics every 2 months, something like that. Yep, they will just fall out on their own.
Does the membrane heal itself?
Yes, membrane will completely heal itself.
But it doesn't do that in adults?
In adults, depending on the size of piercing. I personally know an adult who got the tympanic membrane partially ruptured, and it healed perfectly fine. It just takes time. But if it's destroyed, then it may not grow back completely. That makes sense?
Yes.
Okay.